Good day everyone. In this video, we are going to talk about anticodons and tRNA molecules. You're with me, Lord Mark Aloysius V. Kalangan as your speaker. So in this video, here are its contents. What are anticodons? What are the functions of anticodons? How do anticodons work? What are tRNA molecules? What is the structure of tRNA molecules? And what is the role of tRNA in translation? So for anticodons, these are sequences of nucleotides that are complementary to codons. They are located in tRNAs which allow the tRNAs to put the correct amino acid in line with an mRNA during protein formation. So, during protein formation, amino acids are linked together in a chain, much like beads on a necklace. It is essential that the right amino acids are used in the correct positions since the amino acids have different properties. Putting the wrong one on the spot will make the protein worthless or even toxic to the cell. So here are the functions of anticodons. So the function of anticodons is to combine the necessary amino acids to produce a protein based on the instructions provided in mRNA. So each tRNA holds one amino acid and one anticodon. As the anticodon is correctly combined with the mRNA codon, the cellular machine learns that the right amino acid is in place to be added to the growing protein. So how do anticodons work? So when genetic information is to be turned into a protein, the sequence of events goes like this. The genetic material in the genome of the cell is transcribed into mobile portions of the RNA using the base pairing rules. Each nucleotide has only one other nucleotide that is associated with it. By pairing the right RNA nucleotide with each DNA nucleotide, RNA polymerase produces a strand of RNA that incorporates all the correct details to produce the protein. The messenger RNA or mRNA then moves to a ribosome, which is the protein production site. At the ribosome, the basic pairing rules are again used to ensure the proper transmission of information. Each trinucleotide or codon in the mRNA is paired with an anticodon containing additional bases. The transfer RNAs or tRNA that link proteins together each have one anticodon corresponding to one mRNA codon and one amino acid attached. So when the right tRNA is found to have mRNA, the amino acid is added to the growing protein chain. Enzymes catalyze the attachment of amino acids together as tRNA anticodons bind to the appropriate mRNA codon. So when the amino acid of the tRNA has been added to the protein chain, the tRNA leaves to pick up a new amino acid to create a new mRNA. Next, we have tRNA molecules. Transfer RNAs or tRNAs are molecules that serve as temporary carriers of amino acids carrying the necessary amino acids to a ribosome dependent on a nucleotide messenger RNA or mRNA chain. They serve as intermediaries between nucleotide and amino acid sequences in this way. So tRNAs are ribonucleic acids and are capable of forming hydrogen bonds with mRNA. In addition, it can also form ester bonds with amino acids and can also physically mix mRNA and amino acids during the process of translation. They are combined with mRNA in a complementary and anti-parallel fashion, and each tRNA may be based on an extension of 3 nucleotides to mRNA. So this is the structure of tRNA molecules. tRNAs are encoded by a variety of genes and are usually small molecules about 70 to 90 nucleotides or 5 nanometers in length. The two most important aspects of the tRNA are its anticodon and the terminal 3 hydroxyl group, which can form an ester bond with the amino acid. However, there are other facets of the tRNA structure, such as the D-arm and the T-arm, which contribute to its high level of specificity and efficiency. Transfer RNAs have a sugar phosphate backbone like all other cell nucleic acids, and the position of the ribose sugar gives rise to directionality in the molecule. One end of the RNA has a reactive phosphate group attached to the fifth carbon atom of ribose, while the other end has a free hydroxyl group attached to the third carbon atom. This results in the 5 and 3 ends of the RNA. As all other phosphate and hydroxyl groups are involved in the phosphodiester bonds within the nucleic acid. So the anticodon loop, which pairs with mRNA, decides the amino acid is bound to the acceptor stem. Anticodon loop is identified by amino acid tRNA synthetase or AATS, which is an enzyme that chemically connects tRNA to amino acid via a high energy bond. AATS then reads the anticodon and also identifies the D-arm located downstream from the 5 end of the tRNA. So the role of tRNA in translation is that it is a particle that carries and transfers the amino acid that is specified by an mRNA codon to a growing peptide chain. tRNA particles are single-stranded polynucleotides that contain 73 to 93 nucleotides. Base pairing causes the single tRNA strand to fold back on itself. So, in this representation, hydrogen bonding between tRNA base pairs can be seen through these dotted lines right here. And in conclusion, anticodons are necessary for the protein formation as they are complementary to codons. tRNAs serve as temporary amino acid carriers, and hydrogen bonding is present between tRNA base pairs, and also ester bonding. And that's it. Thank you for listening, and I hope you learned something. Take care.